Good morning, my name is Velalin and my metric number is AN190153. I'm going to present my final year project with the title of The Optimization of Botryococcus Species by Synthetic Growth Stimulant Fertilizer. My supervisor is Professor Dr. Nur Shuhala binti Muhammad Sunar. This is the outline of presentation. First we have Chapter 1, Introduction. Next is Chapter 2, Literature Review, and the third one is Methodology. Moving on to Chapter 4, which is Data Analysis and Discussion, and lastly, Chapter 5, Conclusion. Moving on to project background. So what is the use of microalgae? Microalgae is one of the source of biofuel and food, and also containing high value of biocompounds. Cultivation of microalgae can be beneficial to the wastewater treatment industry and also help with reduction of greenhouse effect and global warming through the reduction of harmful gases such as carbon dioxide. So in this study, we are using NPK as a fertilizer to grow the Botryococcus sp. So NPK widely used as a fertilizer in enhancement of microalgae growth. Next is problem statement. So the Earth needs to reduce the greenhouse gas emission from diesel to stop global warming. So a sustainable biofuel decreases the greenhouse gases emissions and does not have any hindering effect on the local environment in its implementation. So it is good for us to practice this kind of biofuel. Next is green microalgae such as Botryococcus species used in this study because of its high value of biocompounds make it a suitable source of biofuel. Next is NPK help to fasten the cultivation of microalgae. That's why in the study we use NPK as a medium fertilizer to enhance the growth of the Botryococcus cultivation. So the objective of this study is, number one, is to optimize the growth condition of microalgae Botryococcus species in rainwater by using NPK, nitrogen, phosphorus, and potassium. Number two is to analyze the growth rate of microalgae Botryococcus species in 30 days of cultivation period. Next will be project scope. So the research will be carried out using microalgae Botryococcus species that can be found anywhere in freshwater, and the isolated microalgae strain is stored under the microalgae research team UTHM that is ready to culturing and cultivation. And the selective potable water is selected, that is rainwater. So the cultivation of Botryococcus species is done in closed or indoor cultivation by using photobioreactor system in different volume of NPK culture medium, which is blank 0.25 liter, 0.50 liter, 0.75 liter, 1 liter, and 1.25 liter in a rainwater. So the test will be conducted until the growth reaches its peak. It is 30 days with 15 counts of experimentation prior to reach the matured condition of the cultivation growth. Last is Botryococcus species in rainwater will be observed by determining the optical density by using UV-Vis spectrophotometer DR6000 and the cell density using compound microscope and hemocitometer. Next is the significant study. This study is significantly provide an input to our knowledge and understanding the cultivation of microalgae Botryococcus species by using NPK as a fertilizer that will result in more production of microalgae for future green sustainable purposes. Moving on to literature review. So the first part is we're going to talk about the rainwater and rainwater harvesting system. So rainwater is the cleanest kind of natural water since it is created by a condensation of a water vapor in the atmosphere and can be considered as a natural form of distilled water. RHS is the collection of storage of rainwater for human use using a variety of technologies ranging from simple systems to more elaborate constructed systems. Rainwater harvesting has recently acquired popularity as a safe water supply and has been performed since the dawn of time. Moving on, we're going to talk about microalgae Botryococcus species. Microalgae is a subgroup of algae that are unicellular organisms that can survive in both freshwater and saltwater settings. Microalgae require three major components to grow: sunlight, water, and carbon source, and also essential nutrients. Botryococcus species is a genus of green microalgae found in freshwater and brackish water and lakes all over the world, frequently in enormous floating masses. These microalgae, Botryococcus species, may produce a high amount of hydrocarbons, lipids, proteins, carbohydrates, and pigments. Next is cultivation of microalgae. So there are two types of technique of microalgae cultivation. The first one is open cultivation system, such as open pond, tank, raceway pond. The second technique is closed cultivation system that usually use various types of bioreactors. Growth modes of cultivation, there are four growth modes. First one is heterotrophic, the second one is phototrophic, the third one is phototheotrophic, and the last one is mixotrophic. Moving on to chapter 3, and first part of chapter 3 is research methodology. So this is the flowchart of my research methodology. First, we have literature review, and then the next part is microalgae Botryococcus species preparation. The next is cultivation of Botryococcus species via NPK in rainwater. And next, we determine the optical density and the cell density of the Botryococcus species. Next is we determine the Botryococcus species growth rate, and we're gonna discuss the result and moving on to conclusion and the end of the process. Now we're gonna talk about the preparation of the Botryococcus species. So the Botryococcus species were prepared with 17 liters of rainwater mixed together with 1 liter of Botryococcus species for each tank, as you can see from the picture over there. So the medium in this study will be using NPK solution plus rainwater, and the following steps I will talk about is for the medium. So urea 20 gram, TSP 20 gram, and ammonium sulfate 10 gram, measured and poured into the Erlenmeyer flask, and then the Erlenmeyer flask will then filled with water until it reaches 5000 ml. Finally, the aeration needed to avoid dissemination of the ingredients. The preparation of the NPK solution will be repeated to make sure the adequacy of the solution. The analytical balance picture and the NPK solutions are in this slide. Next part is the microalgae growth enumeration. So the growth of the Botryococcus species will observe for 30 days, which is 15 counts, and the optical density and the cell density of Botryococcus species determined every day of testing. Optical density determined by using BR6000 UV-Vis spectrophotometer with wavelength 290 nanometer. Cell density determined by using compound microscope and hemocytometer. The picture of each instrument are shown in the slide. As you can see, this is the observation of day 1, 1 liter of NPK, and day 15 of 1 liter NPK. Next is microalgae Botryococcus species growth rate. So in microalgae Botryococcus species growth rate, there will be 4 phases, which is the first one is lag phase, the second one is exponential phase where the growth reaches its peak. Next is stationary phase where the cell stops to grow, and then the last phase is that phase where the microalgae will start to stop, grow, and death. So this is the equation of growth rate. Moving on is chapter 4, and this is the first part of chapter 4, which is characteristic of rainwater sample. So this is the value that we get from the testing of water quality, which is pH we get 6.4, COD chemical oxygen demand is 0, and biological oxygen demand BOD is 0.68. All of this value we get is still following the National Water Quality Standard for Malaysia. So moving on, this is the observation of the Botryococcus species. So as you can see, there are a huge difference between the two pictures from the left and right, which is day 1 and day 15. We can see that the cells getting more greener at day 15. That indicates that the cell grows over time. Next is for analysis of optical density. This is the graph that we get from the result. So the result shows that the highest growth rate is analyzed to be at 1 liter volume of NPK followed by 1.25 liter, 0.25 liter, 0.50 liter, 0.25 liter, and finally the blank sample. At 1.0 liter volume of fertilizer, NPK, it is only take 8 days to reach the peak, a few days earlier than the other samples. The results show the blank sample still has not reached the peak, even after 15 days. It can be concluded that the fertilizer has a huge impact in increasing and enhancing the growth of the Botryococcus. Next is the analysis for cell density. So, as you can see, this is the graph of comparison of Botryococcus species cell density with a different volume of NPK added. So the result shows that the highest growth rate is analyzed to be at 1.0 liter volume of NPK followed by 1.25 liter, 0.75 liter, 0.5 liter, 0.25 liter, and finally the blank sample. Not much different from the optical density result analysis. At 1.0 liter volume of NPK, it only takes 8 days to reach the peak, a few days earlier than the other samples. The results show the blank sample still has not reached the peak even after 15 days. This is the last part of chapter 4, the analysis for microalgae Botryococcus species growth rate. So this is the graph, the 6 different volume of NPK added, growth rate versus days. From the results, you can see that the growth in 1 liter NPK added reached an early stationary phase at day 9 compared to the other samples that still in growing phases. It indicates that 1 liter NPK is the most optimized culture medium to culture microalgae Botryococcus species. So this is the last part of this presentation, which is conclusion. So the Botryococcus genus is one of the microalgae groups with the potential to solve technology barriers towards economically viable microalgae-based energy generation. Microalgae cultivation requires essential nutrient to grow to keep the cell in exponential phase, where the cell experiences the optimal growth rate. Nitrogen, phosphorus, and potassium are the common micronutrients in microalgae cultivation media. The optimization of the synthetic growth rate was determined by cultivation of Botryococcus species in different volume of NPK as a culture medium, as the optical density and the cell density determined by using UV-Vis spectrophotometer DR6000 with wavelength 219 nanometer and the compound microscope and the hemocytometer. The outcome of this study shows that the Botryococcus species growth rate is higher at 1 liter of NPK culture medium compared to other mediums. This indicates that the optimized medium for Botryococcus species cultivation is at 1 liter of NPK. It is also important to use the right ratio of essential nutrients to maintain the microalgal growth. Excessive levels of nutrient can lead to reduced efficiency in utilization of the nutrient by the microalgae, resulting in reduced growth and also the productivity of the cell. So that is all from me. Thank you.